Good morning and welcome to the 2022 CRB webinar series. Today's webinar is focused on citrus leprosis disease and summer and fall IPM practices. I'm Caitlin Stanton. I'm with the Citrus Research Board, and I'm joined today by McCall Machado and Dr. Joey Mayorkin, who will be running the polls and monitoring the chat. Please note that this webinar is geared toward growers and pest management professionals. Master gardeners can certainly benefit from participating, but the pest management methods presented, especially the pesticides, should not be followed without a clear understanding of their legal use in home environments. With that, I'm going to pass the mic over to Joey, who will introduce our speakers.
Thank you, Caitlin. Please welcome UC Cooperative Extension Area Citrus Advisor, Dr. Greg Duhan. Prior to joining UC Cooperative Extension, Dr. Duhan was an assistant professor at the University of California, Riverside, where his work focused on breeding avocado rootstocks for resistance to Phytophthora cinnamomi, as well as conducting research on the ecology, pathology, and population biology of primarily fungal pathogens. Dr. Duhan joined UC Cooperative Extension in March of 2016, and is continuing to build an extension and research program to benefit citrus growers here in the Valley. Today, Dr. Duhan will provide a regional update. Greg, please go ahead and begin your presentation.
Okay, thank you, Joey, and thank you all for joining this webinar today. Hopefully this will be a thing of the past in the future, because I'm getting tired of them, but that's the way it is. So I think the first thing that I would want to say, I guess, because they wanted me to give some sort of update on what's going on with this last season, maybe the new season. But I was just privy to this like a week ago. There's a new virus called citrus yellow vein clearing virus that they detected in Tulare, and the CDFA, USDA is working on it. Some people may know about it. Like I say, the meeting I had was June 1st. So it was just about a week ago. So that's something to think about. And they're doing surveys trying to figure out what's going on with this virus, how spread out it is. And I think if I remember correctly from the meeting, they only found it in residential. So it's not found in commercial yet, but they're basically on it, and they're basically trying to figure out what's going on. So that's like the one thing that's kind of new. Everything else has been the same. I remember last year when I gave an update, I think it was probably during about the same time. I remember telling people that I didn't have much of a feel for what was going on, because I haven't gotten a lot of field calls about things. And then like a week or two later, there was a lot of thrip issues last year. Some people spraying up to 6 times, which is kind of crazy. But this year, I haven't heard much as well about that. And as far as insect issues, I haven't gotten a lot of phone calls and situations. Over the past couple of years, we have had issues with some scarring on limes and lemons that we quite haven't tried to figure out, or haven't figured out what the actual cause is. And that's something that we're kind of concerned about or looking into. But as far as the regular situation goes, the season, from my understanding, not having these meetings and talking to tons of people, is that it seems like it's a relatively decent season for navels. I've been told that the mandarins were a little bit low, might be an off year, and so next year might be a much better. So all most of the navels are pretty much picked by now, and they're starting to move into the Valencias, and the Valencia crop should be good from all the things I've read about. But as far as like this past year with the diseases, it's always the same sort of thing. It's like usually people call me when their trees are dying, and then once they're in decline, it's difficult to really tell what's going on, because half the time, once these trees are in decline, they get Fusarium solani, which is a secondary pathogen, and they come in and just take over the tree once it's already sick. So what was the original reason why this tree was sick? Was it some sort of incompatibility, crazy weather, not enough nutrients, not enough water? it's always hard to work backwards multiple years to figure these things out. And then once again, the same sort of issues, all the dieback stuff everybody's pretty much used to dealing with through all the years. You have to go out and, prune out all the dead wood and get it out of the field so you don't infect the trees the next year and all that stuff. So that's pretty much it. I can't think of any major issues other than this new virus, which I'm actually tomorrow going to go out and actually take a look at a place where they have trees that have symptoms, so I can actually physically see it myself. I've seen pictures of it, but I haven't seen it. So that's pretty much it, I think, for a sort of a wrap up. Does that sound good?
Okay, that sounds good, Greg. Thank you so much for sharing that update with us today.
Okay.
Please welcome today's speaker, Dr. Sandipa Gautam, with UC Cooperative Extension, who is the UC Cooperative Extension Area Citrus IPM Advisor. Prior to joining UC Cooperative Extension, Dr. Gautam was an assistant research entomologist at the University of California, Riverside, where her work focused on managing citrus pests that have export significance. In her current position, Dr. Gautam continues to focus on the integrated management of insects and mites that are important to California citrus production. Today, Dr. Gautam will discuss two topics of interest. The first part of the talk will introduce citrus leprosis disease, its identification, and its effect on citrus. The remaining portion of the talk will focus on pest management practices for summer and fall, and will focus on California red scale, Citricola scale, fuller rose beetle, and citrus mealybug. Sandipa, please go ahead and share your screen to begin the presentation.
Thank you, Joey, for that introduction. Okay, thank you, Joey, for that introduction, and thank you everyone for joining today's CRB webinar series. And as Joey said, I'm going to focus on two different topics today, introduce you to this new emerging viral disease of citrus that is called citrus leprosis, and talk about summer fall citrus IPM practices. Here is the outline that I'm going to follow. We'll talk a lot of things about citrus leprosis, introduce the disease, talk about where the disease is currently present and what's the timeline that this disease is spreading into newer citrus growing regions of the world, talk in detail about disease symptoms and the vector and some disease control methods. Then after that is done, we'll switch gears and talk about summer and fall IPM practices, talk about the best management practices in summer and fall, what you can do, what you should be doing around this time of the year, and introduce you to another emerging topic, which is citrus mealybug in the San Joaquin Valley. So what is this disease? What is citrus leprosis? It is a non-systemic viral disease of citrus caused by the virus that's named citrus leprosis virus. What do I mean by non-systemic? I mean that the disease do not move from one part of the tree to another part. It has to be transmitted by mite, and it only develops in an area where the mite has injected the virus. These viral agents cause chlorotic and necrotic lesions on citrus leaves, fruits, twigs, and branches, and the lesions only develop in and around the area where the mites have fed upon. And as lesions grow, these lesions could coalesce together and cause premature leaf and fruit drop. There are several different viruses, 12 to be exact, belonging to 3 different genera that can cause symptoms that are similar to citrus leprosis symptoms. It's not important for you to know all of these, but be aware that there are 2 different kinds: cytoplasmic type, that is commonly known as CILVC, which affects the cell, and virus multiplies in the endoplasmic reticulum of the cell. It is the most common one, and it causes bigger size lesions compared to the nucleus type, which colonizes the nucleus of the cell and causes smaller type of symptoms. Viruses are transmitted by Brevibulbous mites, commonly known as flat mites, and we'll talk more about these mite species later on the talk. So why is citrus leprosis a rising concern? In the last several years, citrus leprosis has been reported many times, a lot of studies published, and it has been talked about the most devastating emerging viral disease of citrus. This disease has always been present in Brazil, and Brazil every year spends more than $50 million to manage this disease, to prevent crop from dieback and premature fruit dropping, which, if not controlled, could cause up to 100% fruit loss. In recent years, this disease has been reported in other areas of the world, other citrus growing regions, and it's rapidly expanding, which is why it's becoming more of a concern. And all citrus varieties, including the rootstocks and cyans, are affected. And what is even more concerning is that it is transmitted by a vector, mite, which is very small, very hard to see with naked eyes. And the vector can colonize up to 40 genera of different plants. So it's found in many different plants, including citrus. And to date, there is no cure for the disease. Let's look at where the disease is present. This map shows the yellow circle, so where the disease is currently reported to be present. And as you can see from the map, it's pretty much everywhere in South America and Central America, and it's making its way to North America. It was reported in Mexico in 2005, and recently in 2020, it has been reported from Hawaii. Another incident reported in 2019 from South Africa shows that it's crossing continents, and now this disease is found in 3 different continents. In the United States, this disease was present in Florida, was reported in Florida from 1860s, and last reported from Florida in 1968. And since then, there has been no other reports of citrus leprosis disease from Florida. It is thought to be eradicated, and the disappearance of the disease from Florida is believed to be caused by higher use of acaricides to control mite species and freezing weather conditions in Florida that killed a lot of citrus trees and may have killed vector mite populations in the 1960s and 70s. Talking about the United States, in 2020, this disease was detected in Hilo, Hawaii on rough lemons and mandarins. So this disease has been in the United States and is coming to the US. Just a little bit more on occurrence and spread of the disease through the Americas based on the timeline table that I have taken from a publication in 2021. Childress and Rodriguez developed a timeline of where the disease was present and how it has been reported over time. If you look at the table, in 1920s, the disease was reported from Argentina, very south of the South America. And in 1933, the disease was found in Brazil, one of the biggest citrus producing countries of the world, and has established in Brazil since then. And like I said in my earlier slide before, Brazil spends about $50 million per year to manage this disease. Later in 1995 and 2000s, the disease was reported from other neighboring countries in Brazil. And by 2005, it was reported from all Central America except in Belize. And in 2005, 7, and 9, it was reported from different parts of the Mexico. Now, after this study was published in 2021, the disease was reported from Belize and recently from Hawaii. So this disease is pretty much present in all parts of South America, Central America, and it's half of North America. It's present in Mexico. What does it attack? Which plants are the host for citrus leprosis disease? As the name suggests, all the plants in Rutaceae family are host to this disease. All rootstocks and cyans are known to be affected by citrus leprosis. Sweet oranges, mandarins, and their hybrids and grapefruit are reported to be the most susceptible varieties. And these three varieties are also the most popular citrus varieties commonly grown in California. Other citrus varieties that are known to be affected are lemons, limes, rough lemons, citron, Mexican lime, and sour orange. And many other wild relatives of citrus are also known to develop disease symptoms. Because there are several different viruses, 12 different strains of virus affecting citrus and causing similar symptoms as citrus leprosis, it is also in recent years been reported from other host ornamental plants, to be exact. It has been reported from hibiscus species and Diphenobacia, and in orchids it causes orchid flake virus. The incidence that was reported in Hawaii is in fact caused by orchid flake virus, which is a type N virus causing citrus leprosis in citrus. So what it says is pretty much everything citrus can be affected by this disease. What happens when the citrus is affected? What do the symptoms look like? So it is important to remember, and I'm going to keep coming back to this, that this disease is transmitted by a mite species, and it can only be transmitted by a mite species. The disease symptoms develop around the area where the mite has fed, and the disease symptoms is the virus establishes and affects the cells, the disease symptoms become enlarged. And as you can see from these pictures, each of these spots is an area where a mite has fed and introduced the disease. So that circle is one mite feeding, this circle is another mite, this circle is another mite feeding. So once the disease starts developing, what happens on leaf is the leaves start changing color and you see these chlorotic spots. And if there is a lot of mite feeding, these spots could grow and the two lesions can coalesce and the whole leaf could look yellowish, like, or a bigger portion of the leaf could look yellowish. That's how the symptoms on leaf look like. Now, these symptoms on leaf are flat or sunken. These are not raised. If you feel it, you cannot feel any coarseness on the leaf. The leaves feel smooth, but what you can see is yellow halo around the feeding spot of a mite. On the stem, disease symptoms look a little different. Here also, the symptoms develops from the area where mite feeding injury, mite has fed and injected the virus, and the lesions develop and become larger and could merge. And if that happens, it leads to the death of the part of the stem. On leaves, the symptom is smooth, but on twigs and branches, the symptom is somewhat flaky. Once the tissue starts dying, the outer bark of the cell starts coming off, and it could give a flaky appearance when you look at it, and you could feel the flakiness when you feel it. What happens on fruit? Because the mite also feeds on fruit, when a mite brings the virus and injects the virus onto the fruit on a feeding site, the symptoms will start developing. The picture on the left is the early symptom of citrus leprosis on immature fruit. As you can see, the chlorotic lesions circle the point of the vector feeding. It starts becoming yellowish in color and grows in size. And over time, what happens is that center of the tree, center of the feeding damage would start turning necrotic, meaning the cell will die, but it will still have that yellowish halo around it. And severe symptoms on the fruit are the photos on the very right. As the disease progresses, the symptoms becomes more necrotic and the fruit looks like it has been hit with God knows what. And citrus leprosis disease has also been known to advance the maturity of the fruit. And if, the disease symptoms are around the area where the peduncle is attached, it leads to premature drop of the fruit. And severe infestation can destroy the orchard. In orchards where citrus leprosis is prevalent, there is a lot of fruit drop going on. Secondary pathogens can affect the fruit, again, leading to fruit drop and rot and whatnot. And when the disease is present on leaves, it could lead to branch death and eventually tree death if the block is not treated. Now, how does this disease get around? If it's only transmitted by mite, and it is only transmitted by mite, the disease gets around along with the vector. It is the vector Brachypulpus mites, and there are several species of Brachypulpus mites that are known to transmit citrus leprosis disease: Brachypulpus californicus, Brachypulpus ovavatus, Brachypulpus eutherosii, and Brachypulpus finesis are the four known vectors of citrus leprosis disease. And the disease moves and disperses along with the vectors. Once acquired, the virus stays in the mite body, and it can infect your feeding sites as long as it lives. And Brachypulpus mites are very common mite species that colonize many citrus species and many other crops. The leprosis disease is not systemic and is not transmitted by seeds. It does not move from one part of the tree to another part. So if you graft a budwood that has citrus leprosis lesion, but you don't have mites in your block, then that disease will stay localized on that budwood and not move to the other regions of the tree. It can only move short distances on grafted tissue, but if the vector is not present, the disease does not move. A little bit more about vector. This several species of Brachypulpus can vector citrus leprosis disease. Brachypulpus mites are very small mites that are about 200 to 400 microns in length. Cannot be seen with naked eyes. You would need 20x hand lens to see them in the field. They have flat body, somewhat oblong sepate body, and depending on what they are feeding on, their color could be orange to reddish brown. The mites can acquire and transmit virus throughout their life. All feeding stages or all live active stages, adult, larvae, protonymph, which is the first nymph, and deutonymph, which is the second nymph. All of these life stages can acquire virus and transmit virus when they're feeding, and all these life stages actively feed. But this disease is not transmitted from female to eggs. In order to get the virus, they have to feed on the lesion. Life cycle of the mite is completed in about 3 weeks, and adults can live up to 3 weeks. So there is about 6 weeks when these mites can pick up the virus and continue feeding, and they can live longer, especially if the temperature is lower. And there are several species of Californicus that can transmit transmit the citrus leprosis disease. Californicus ovavatus, Phoenix, and Etherosi have been confirmed as vectors, and several other species are being researched for their ability to acquire the virus and transmit. There are some species which can acquire the virus but not transmit, and some other Brachypulpus species cannot acquire the virus. Why is it a concern for us, and why are we talking about it? Because Brachypulpus mites are present in California. It infests citrus. It's a minor pest in coastal regions and desert regions. There are 40 genera of plants that are host to flat mites, so it's pretty much everywhere. It's probably present in your backyard. It is a warm season pest. It's usually active around this time of the year and is active through September, October, and slows down during the harvesting season. It likes to proliferate in an area where there is feeding damage caused by some other pest. For example, in this picture on the top right corner, you see this orange spots. All of those, that's probably 50, 60 eggs in that cracks. They are that small, and they like to lay eggs in cracks. And eggs, when they hatch, they feed around that area, and move on and, infest in new area. They are, if we're talking about citrus, the mite species found in citrus blocks, they are smaller than other mite species that we usually see in citrus. Citrus red mite is about 0.5 millimeter long and has long hairs. So you can easily tell citrus mite, they move very fast. Two-spotted spider mites are rather spider mite species that are commonly found in citrus. You can identify them by the two spots they have on the lateral sides of their body. They also are much bigger in size and move fast. Braypulpus mites are comparatively smaller, half as small as red mites or two-spotted spider mites, and have a flat body. It would look like that thing is attached to the leaf, but it has legs and it moves very, very slow. Those are a few of the key characters that you could tell Braypulpus species from other mite species. There haven't been any field studies done on Braypulpus mites showing efficacy of miticide, but we tested several different chemicals in lab, and most of the acaricides registered in California are very effective in controlling Braypulpus mites in laboratory studies. Okay, so a little bit more about why is citrus leprosis virus a concern for California citrus. If you're aware with the disease triangle, there are three situations that are needed for a disease to proliferate, disease to establish in a new area. Host, which is citrus in this case, which we have a lot in California. Environment, which is combination of temperature, humidity, weather conditions, and in this case, a vector, which we have in California. What we don't have is a pathogen of citrus leprosis. And once that pathogen gets here, because we have other conditions that completes the disease triangle, it is very likely that the disease will establish in California, which is why it is a big concern for California citrus, and it is important for us to be aware that this disease is an emerging threat to fresh citrus here in California. What can we do about it? Prevention is much easier, cheaper, and better than cure, because California's climatic conditions are highly suited for the establishment of disease, and the vector is already present in California. The risk is high, so preventing leprosis from entering California is much easier to achieve than trying to eradicate it once it's detected. And how can we do that? We can do that by avoiding, regulating the transportation of propagation material that may either have citrus leprosis lesions, or mites could come from another country. And because of the HLB and regulations that are in place, I think we're already doing a lot of that. And it is also important to know that this disease can only be controlled by controlling vectors, and knowing about how the mites, where the mites are present, how the mites look like, is another, and being educated on mite damage and identification is another way to be vigilant and be on the lookout for when this disease may come to California. And if you suspect citrus leprosis in your grove, if you think the lesions on your lesions that you're seeing on your fruit or your leaves look like citrus leprosis, please contact your local county ag commissioner or extension specialist for diagnosis. I can help, Greg Duhan can help, and there are other UC researchers working in California who can help diagnose the disease. That is all I have for citrus leprosis. We're going to move on, and I'm going to switch gears and talk about summer and fall IPM practices, and we'll focus on best management practices in summer and fall for three key pests: California Red Scale, Citricola Scale, and Fluorosphere Beetle, and also spend some time talking about citrus mealybug and how much do we know about it, and how concerned are some of us regarding this new emerging insect. So my focus for this part of the talk is going to be Sandokan Valley citrus IPM. Traditionally, pest management in the Sandokan Valley in citrus has focused on key pests that are mentioned in this slide. These pests are categorized into two different types. The ones that cause cosmetic damage usually come early in the season, affect the fruit very early in the fruit development. Those can be citrus red mites, citrus thrips, and katydids. And the other pests usually causing yield damage and also cosmetic damage if the infestations are high are California Red Scale and Citricola Scale. California Red Scale is a sap-sucking insect. It attacks all parts of the trees, including fruit, and it downgrades the fruit by just being present on the fruit, and causes yield loss and causes dieback and tree death. In the last decade or so, California Red Scale has been really established in many parts in the Sandokan Valley and is becoming harder and harder to control. Let us look at the life cycle of Red Scale and talk a little bit about how we can use this knowledge about life cycle to make pest management decision. So Red Scale starts the seasonal cycle of California Red Scale starts as overwintering gravid females. These gravid females have eggs in them, and as temperature rises, the eggs start hatching and the crawlers move out. And the crawlers, when they come out, they are the only moving life stage of California Red Scale, and they move a few inches, go onto the new tree, or gets blown away by wind to a newer area. And what happens is they start feeding. They insert their stylet, start feeding, start producing the wax, and cover their body with the wax, and turn into what we call white caps. There are two white caps in the pictures that you can see below. And then they go through the first molt. Until the first molt, you can't really tell between male and female, but these then molt once. Females then molt once and become second instar, and then they molt again and become third instar. Third instar female is what you are seeing in the picture on the top right corner. The female is yellowish, lemon yellowish in color, and has lobes on the side, is plump looking. This is a healthy female in this picture. And if you look closely on the arrow, there are two molting rings. So females molt twice. Males molt multiple times. They go through second instar, then go through another molt, become a prepupa, then go through another molt and become pupa and adult. Now, males, when they emerge, their only job is to find a female mate, and they are very short-lived. So they find a female mate, and they mate with third instar females. And when mated, these third instar females become gravid females. And gravid females are the ones that produce eggs, and the generation completes and continues. For Red Scale management, PCAs have long relied on monitoring the populations and predicting when different life stages are occurring in the field and when different generations are occurring. Red Scale usually has 4 generations on when the weather conditions are nice, but if it's hot and dry weather conditions with high summer temperatures, it could have as many as 4.5 generations. In Kern County, they have also seen 5th generation crawlers, but it doesn't complete the 5th generation. So how do we monitor it? There are several ways to monitor for Red Scale population. You can monitor for males by using pheromone trap cards. Pheromone trap cards are the ones that are shown on the top right corner here. What pheromone cards are put out very early in the season, beginning of March, and then changed weekly to figure out when the males are flying, then that's going to tell you when the generations are occurring. There are usually, there is a big peak on the first flight, and then second and third peaks that you can probably tell. And also fourth flight is used as an indicator to tell if your grove will have red scale populations in the following season. And male trap cards are also important because based on the biofix, which is when the onset of male flight happens, then successive generations are predicted based on degree day accumulation on when certain number of heat units are accumulated for the insect to complete the next life stage, and when the next flight is going to happen, or next generation crawler is going to emerge. We'll talk a little bit more about this in the next slides. Once you start seeing males and you have your degree days and you know when the crawlers are going to emerge, you can confirm that by using crawler tapes, which is another method usually used by researchers, because we want to make sure that we're doing right timed application when we're doing efficacy trials on insecticides. Crawler tapes are targeted against crawlers. What you do here is you find a twig that has a female scale, and you wrap sticky tape around the twig and change that every week for a couple of weeks, and count the number of crawlers. If you find crawlers, then you know that the crawlers have started to emerge, and they will settle and become white caps and molt. Another common method that you can use is you can walk the block and you can sample leaf and twig, and you can do this for all life stages during the season. You pull a leaf and you look at the leaf or a twig, and you can tell dead and live scale by rubbing your fingers on the leaf. The dead scales are flaky and will fall off. The live scales will not fall off, they will stay in there. And another way to monitor for red scale population, especially targeting the following season, is looking at fruit at harvest and determining what percentage of the fruit at harvest has red scale, and estimate the percentage of fruit with more than 10 scale. And if your block has more than 5% of the fruit infested with red scales, then you might need a treatment in the following season. Degree days is a very useful tool and a very important concept to understand, especially for red scale management. All insects develop based on heat units accumulated above their lower developmental threshold. Insects are insect development is temperature dependent, and they can only develop once that temperature threshold is reached and we have high heat units accumulated above the lower developmental threshold. What you're looking here in this slide is males per trap card, the data for males per trap card, first generation peak male flight early in March, 13th of March, and after that 550 degree days after that, you can expect crawler emergence. It takes that many degree days for California red scale to go from egg to crawler, and 1100 degree days after the first male flight, you can expect another male flight, and so on. How can we calculate degree days? It's a pretty simple to calculate degree days. The lower developmental threshold for red scale is 53 degree Fahrenheit, and any temperature accumulation above the LDT counts towards DD or degree days. So let's look at two different scenarios here: spring temperature and summer temperatures. So in this case, we have a high temperature of 77 in spring, low of 75. So the average deadly temperature is 61, which is high plus low divided by 2. So the degree days accumulated in spring is 8. the average daily temperature minus the 53, which is lower developmental threshold for California Red Scale. So in this case, number of degree days needed from males to crawlers is 69 days. That's a long time. But when it's hot and we have a high heat, high temperatures and high low temperatures, we have much higher degree days accumulated. And in this case, 41 degree days accumulated. And when that happens, the 500 degree days required from males to crawlers can be completed within 14 days. So things are happening really fast in summer and slower in the spring. And we do the calculations for you and update the information on Lindcove website. Here is a link for the degree days in 4 different counties. And this is the current degree day accumulation in these 4 different counties. We already saw first male flight in all these counties. Kern County, as of June 5, has 1,137 degree days accumulated. So you can expect to see crawlers in this week or in the coming weeks. Tulare is a little behind, and it may take another few weeks for the crawlers to start emerging. So you have about 4 or 5 weeks from now in Tulare County and Fresno and Madera County for the crawlers to emerge, and we'll continue to monitor this. So you can check how many degree days have accumulated in these different counties on the LRIC website. That being said, degree days also depends on where you're located and also the orientation of your orchard. So if you are relying on degree day models to predict populations and make your pest management decision, it's a good idea to have a temperature recorder in your block and monitor for degree days for the particular block that you are concerned about. Let's talk a little bit more about why we have so many Red Scale and why it's becoming hard and hard to control. In the last decade, we have had warm winters, and what that means is scales are developing at all times, and there is less overwintering mortality. So we have high scale populations early in the season. Red Scale likes dusty stressed trees, and they do better in those trees, and also parasites don't work very well on these trees. So avoid water stress to the trees because stressed trees are vulnerable to Red Scale. Another reason is because of the high heat, there is fast development of scale happening, especially in the Kern County. As you can see in this graph, the black line, it was supposed to be dotted. I don't know why it shows as a continuous line. The black line is a 30-year average cumulative degree days. And the 2 lines below the black lines are 2010 and 11, which were the cooler years in the San Joaquin Valley in Kern County. And the last 3 years, 2020, 21, and 22, are the purple, blue, and red line above the black line. As you can see, there is, things are happening faster, and there is a higher number of degree days accumulated for a 5th generation flight in Kern County. Also, it's becoming hard to control because insecticide treatment only lasts about a generation, and there is not a lot of insecticide products. It forces growers to treat more, and that breeds resistance problem. And some insecticides don't work on scale, and the scale populations builds on wood and serves as a reservoir to infest fruit and leaves in the next coming season. So what are our choices? What choices do we have for Red Scale management? There are several. We have 7, which is moderate in efficacy, but has resistant issues. 7 is toxic to most natural enemies, but it's a broad spectrum, and it will work on Citricola scale, Fluorosphere beetle, as well as on California Red Scale. Next two on the list are steam and center. These two are insect growth regulators. They are they only target red scales, so are selective, but are toxic to beetles because they are insect growth regulators and also can affect beetle populations. Movento is another insecticide which is heavily relied on to control red scale, but it doesn't work on scale on the wood. It's very effective on the scales on leaves, but it doesn't work on scale on woods, and it's predatory on it's toxic to predatory mites. Also are moderately effective if you can cover things in oil, but if anything oil doesn't touch, it doesn't kill. It has a short residual and it has short-term effects on all arthropods. It's a broad spectrum. It will kill anything that will that is smothered in oil. The two safest methods are aphides, and aphides is a really good biocontrol. It's moderate. It cannot eliminate the population, but it will do a very good job, especially if you choose softer chemistries and are aware of aphides population when making your pest management decisions. Mating disruption is becoming really popular and is integrated as a part of the citrus California Red Scale IPM. It's moderately effective, and for it to effectively influence the population you need to adopt the dispenser into your IPM program for multiple years. It's not going to work in the first year. You probably need to apply the mating disruption for successive 2 to 3 years to see any effect. It's non-toxic to any other pest, and it is narrow on it has a narrow spectrum and only works on red scale. Now, let's go back to biology and talk about how to use life cycle information for making pest management decisions. Savin and all are harsh chemicals, and it's most effective when applied on whitecaps and crawlers because they are the most susceptible stays. They don't have that protective waxy layer covering their body. So it's most effective applied early in the season. Steam and center are insect growth regulators, and those need to be applied before insect completes the molting because they affect molting into the next life stage. So in any generation, if you're applying steam and center, do it when the crawlers start emerging. Movento works on all life stages, but it only affects red scale on leaves and fruit. And if you're releasing aphides as a part of your red scale IPM, aphides only affects 13 star females. So release them around, time them in a way that 13 stars are present in your block. And mating disruption needs to go very early in the season after the trees are harvested and pruned. It's better if mating disruption can be applied in the blocks before the onset of first generation flight, if not before any time between first and second generation male flight. When to spray, making decisions. PCAs have relied on pheromone cards to watch populations and made spray decisions based on how many numbers you're catching and based on degree day calculations of when the next generations are emerging. But that pheromone cards and how to watch populations and degree day models and the numbers especially were developed in an era where organophosphates and carbamates, the broad spectrum insecticides, were very popular, and they can still be a very reliable indicator of populations in the field, but the numbers you can rely totally on how many males you're catching per trap, because there are two different scenarios. If you're using insect growth regulators and pheromones, you could be catching female scales on the card but have more scale populations, because insect growth regulators affect males more than it affects females because males molt multiple times, and pheromones affect males only. So if you're using those two, you can have more scales in the block, but less scales on the pheromone cards. In an adverse scenario, if you're using a Phytis and Movento, you can actually have more males and less scale population, because a Phytis affects female and Movento will kill pretty much everything that's on the outer canopy of the fruit, and you're probably catching more males from the ones that are developing on the wood. So in addition to pheromone cards, also leaf twig sampling is helpful to make spray decisions. It's important to look where the scales are, sample as if it's coming from your neighbor, you'll probably find more on the border rows. And if you're finding a lot of scales on the trap cards, but you're not seeing scale on the lower part of the tree, look on the interior and top, because sometimes insecticide applications don't reach up interior part of the tree and up top, and scale populations can develop there. Also determine if the scales are live. Rub your thumb lightly over the scales. Scales don't usually flake off if they stay there. They don't come out until the external force is applied. So to determine if the scales are live or dead, you rub your thumb lightly on the scales, and if all of the scales flake off, you did a pretty good job of controlling them, and you may not need to make another application. And after each application, it's a good idea to evaluate how the treatment affected, and count the number of dead, healthy, and parasitized scales to determine how much Phytis parasitism is helping. And another practice you can do in this season is plan for the next season. And that you can do by using pheromone trap cards. If you rely on pheromone traps, monitor fourth flight. The traditional number was 1,000 males per traps. Then if you're catching 1,000 males per traps, then you would need a treatment next season. But things are changing, and if mating disruption is a part of your IPM, I have learned from growers and PCAs that 50 males scales per trap is enough enough population that you may need to treat in the following season. Also planning for the next season, scout for fruit infestation with red scale during harvest, and count the number of fruits that has more than 10 scales. And if 5% of your fruit is infested, it may warrant treatment in the following season. Let's talk a little bit more about mating disruption. What is mating disruption pheromone? How does it work? Mating disruption basically is putting out so much pheromone produced by female out in the environment so that we are confusing the males and they can't find females. And what happens if they can't find females? Female scales, how do females behave in the presence of pheromone disruption? Males, their only job after they emerge is to find a female and mate. And if they can't find a female and don't mate, the females, if they never get mated, they don't produce crowders. They stay as third instars. If the females mate late in life, they produce fewer scales. If the females are mated late in life, it's been shown that they produce mostly males. And if they remain third instar, they're more vulnerable to Phytis. So there are many ways that mating disruption can help lower red scale population in the block. What this graph is showing out here is just confirming if females don't get mated, they remain third instar from the data. What Joel Leonard, master's research student, did in this study is he put out sentinel insects in the grove where mating disruption was applied and in the grove where mating disruption was not applied, and he brought those back to the lab and counted the number of third instar females and adults. And as you can see, where the pheromone was present, in the block where pheromone was present, they did not become adults. But in control, where the pheromone disruption was not present, they became adults. Most of them already became adults in week 1, and all of them became adults in week 2. So again, going back, that if they don't get mated, they remain third instar. Also, some more data from the study that Joel Leonard did for his master's thesis, where they studied mating disruption at the rate of 180 dispensers per acre in 2 orchards in 2016 and 17, and 10 orchards in 2018 and 19. In 2016, the traps were put out after the first male flight, and in 2018, the traps were put out after the first male flight. And what this data is showing to you is males caught per trap on the y-axis and the time in timeline in x-axis. In both of these studies, they found that there were, you can see these peaks for first male flight, second generation male flight, third generation male flight, fourth generation, fifth generation male flight in control, but in mating disruption, where the mating disruption was applied, very few were trapped on the trap card. And when they looked at percentage fruit infestation in these different treatments, they found that in control had higher percentage fruit infestation with Red scale compared to where the pheromone disruption was applied. So when all these numbers were crunched and all the data was analyzed, what it means is if your male capture on trap cards is reduced by 95% compared to the control, or compared to what you were catching in earlier years, then you'll get more than 95% reduction in highly infested fruit. Now you ask, how do I know if I have 95% reduction of highly infested fruit? That you can confirm by sampling. If you look at 5 twigs, at least with 5 leaves in August, and count the number of mean life scale per twig, and if you're finding you have 1 or more life scale per twig, then you're going to have scaly fruit at harvest. If you're finding less than 1 life scale per twig, then you can say that you have 95% reduction in red scale population in your block where pheromone disruption is applied. Okay, that's all about California red scale. Now let's move on to Citricola scale, which is another issue for citrus growers in the valley. Citricola scale is not a problem every year, but it could be a problem in cooler years, and it could be a problem that's developing over the years and could blow up randomly in a year. So let's start with Citricola scale life cycle. Put out on a calendar, annual calendar. This is how it looks like. Citricola scale have only 1 generation per year, but they're very, very prolific. So what we start with the year with is second instars on twigs. They miraculously molt and become adults, egg-laying females in March and April, and start laying eggs from April through July. 1 female could lay up to 3,000 eggs, and these eggs hatch and become crawlers, and these crawlers move from the inside canopy of the tree where they're present to the outer canopy of the tree, and you'll find first instar females on the outer canopy of the tree on newer leaves. And late in August or September, they start molting, and the second instar, once it starts cooling down in October, they start moving to the inside canopy of the tree. Sometimes these different life stages do not overlap because it only has 1 generation per year, but knowing about the biology affects the timing and sampling of the treatments, because we want to apply treatments to the most vulnerable life stage, and that is first instar nymph, because their scale covering the body is not tough as second star or adults, and also because they are present on the outside canopy of the tree. How do we sample for Citricola scale? We recommend sampling in July and August when most of the eggs have hatched and crowders have moved out to the outer canopy of the tree. What you do is you walk up to the tree, go to the northeast corner of the tree. It's usually the cooler side of the tree. Citricola scale likes cooler temperature, and pick a leaf, randomly pick a leaf from a tree and look at the leaf and count the number of live first instar nymph. And randomly, keep doing that in 25 trees in a row, and then do that same process in four different rows. And if you have 0.5 scales per leaf, that means one scale for every 2 leaves that you looked at, then you have a Citricola scale situation, and you need to be making an insecticide application. I talked about identifying live scale, and it is important. There are several other factors that could kill Citricola scale. Oil treatment for other insecticide may kill Citricola scale. They could die due to natural causes. They could be fried by the high heat temperatures. So it is really important to determine what percentage of live scale infestation you have. And how do you tell a live scale? Live scale is more plump, it's translucent in color, and one of the key identifying characters is you can see the legs. If you're looking at the picture on the top left corner, you can see pairs of legs going in 2 different directions, making somewhat H shape across the body. If you can see that when you're looking at your first instar nymph, then that is your live scale. You can't see the legs in a dying or dead or a flaky first instar nymph that's dead due to natural causes. So that's one of the key identifying characters for telling live first instar nymph. I took these recommendations from UC Ipm guidelines. There are several products that are listed for controlling Citricola scale. Center is insect growth regulator. We have SL, Actara, Sevanto, Malathion, and 7+ that will control Citricola scale. Also want to say it here that while no longer registered for use in citrus, made for many years, Lorsban was a standard product to control Citricola scale, and recommended time for application is late July, early September to target the most susceptible life stages. You want to wait until most of the scale nymphs are in the outer canopy of the tree, and that's usually late July, early August through September. Next insect on the list is Fullerose beetle. And we're talking about Fullerose beetle not because it is a pest of citrus, but because it is an export concern pest in South Korea. And the regulation require that field treatments be applied to control Fullerose beetle in field. Fullerose beetle is also an insect that only has one life cycle per year. So adults emerge pretty much throughout the year, and they feed on leaves and they lay eggs under the sepals, which is why it's a big issue in Korea. That's their preferred egg laying spot. The first instar larvae takes about 4 to 6 weeks to hatch. Could take longer if the temperature is lower. And the first instar larvae, when it hatches, drops to the ground and feeds on roots and other things under the ground, and goes through several molting stages for about 9 months under the ground. And it pupates, and adult emerges and climbs the tree, and the life cycle begins again. Now in the Sanokan Valley, adult emergence starts picking up in June, and highest emergence have been recorded in August, but it does emerge throughout the year. And what that means is foliar pesticide treatments are difficult because of the year-round emergence. How can you tell if you have Fullerose beetle in your grove? You can tell by observing the leaves for feeding damage. And also, these are big insects. They make a lot of frass. If you're seeing these frass pellets on your leaf, then you probably have a full rose beetle situation in your block. About the feeding damage, full rose beetle like feeding on the leaf, but they go around the leaf and notch the margins of the leaf. So you have this ragged looking leaf on your block. If you see this is a characteristic damage caused by full rose beetle feeding. Another way you could sample for full rose beetle is by using a beating seed. Take a beating seed and a stick, and put the seed under the tree and just tap on the branches, shake the branches. The beetles will fall down and play dead, and you can quickly count how many beetles fell out. If you find few beetles, you can apply insecticides, but if you find 10 beetles fall out while you're beating a tree, then avoid sipping that Fruiticoria because you probably have eggs under the sepals. Late in the season, during harvesting, you can also sample for eggs, and you can do that by twisting the calyx. And if the eggs are there, you're going to see these yellowish, whitish plump. They usually lay eggs in clusters, and it's cemented to the calyx. So those are the ways that you could tell if full rose beetle is present in your grove. Current regulations for controlling full rose beetle are scud pruning, because these beetles are flightless. They need a pathway to climb up to the tree when they emerge from the ground. And if you scud prune the trees up to 30 inches height, then that prevents beetles from climbing up to the tree. Also, control weed under the canopy of the tree so beetles don't have anything to taking them up to the tree canopy. And 2 treatments are recommended, either applied to the ground or foliage. Scud prune is usually recommended early May or late April. First treatment before the peak emergence happens in July. Usually first treatment recommended is a soil treatment because you're trying to prevent adult emergence and kill those pupae and late stage larvae in the soil. Second treatment is recommended sometime between September and October. So you're preventing beetles from laying eggs before the harvest. Timing of application also depends on grove location and when you're going to be harvesting your plot. Now, let's move on to the last insect of today's topic, citrus mealybug, and what we know about it. Citrus mealybug is a rising concern in the Central Valley in the last 2 or 3 years, especially in the last year. It has been there has been multiple reports from various species from Fresno County, Madera County, multiple in Tulare and Kern counties. It is reported from many different citrus varieties. Navel oranges and lemons are the most common. Lemons more so because this bug likes cluster of fruit. How does it cause damage? It is a sap-sucking pest. It attacks all parts of the trees, including fruit. It produces a lot of honeydew and a lot of sooty mold and could really affect the quality of the fruit, like you see in this picture. Heavy feeding can reduce tree vigor and cause fruit drop and fruit contamination by sooty mold or mealybug can affect the marketability of the fruit. What do we know about its biology? We know a little bit, but we don't know a lot about what's happening out in the field and what the seasonal biology and seasonal phenology of this pest is in the San Joaquin Valley. What we know is this pest has both males and females. Males are very short-lived. They emerge, mate, and die. Mated females lay eggs in egg sacs that is loosely held by silk threads and covered in mealywax. They could lay up to 200 to 300 eggs. One single female can lay up to 200 to 300 eggs in their lifetime. And after the egg laying is done, they shrivel and die. These eggs hatch and the crawlers disperse and feed and move to different part of the trees. Ants, especially Argentine ants, have been known to farm mealybug and literally carry mealybug prowlers and adults from one place to another. So it probably losing chlorpyrifos as an ant control method is one of the reasons why we are seeing mealybug in different areas in the San Joaquin Valley. Now, how do you tell if you have citrus mealybug in your grove? It's really hard because unless the populations are established, because the population starts as a spotty infestation, it's difficult to monitor. But if you know that you had mealybug in an area and identify hotspots, you flag those areas and monitor through the season. One of the ways to do it is look for sooty mold on the leaves. If citrus mealybug is present, they will be there. Other insects producing sooty mold and honeydew in San Joaquin Valley are Citricola scale and Cooperative scale. You could also look for those. They are not that big of an issue, but citrus mealybug could look this bad as shown in this picture. There are pheromone traps, and if you trap males on a pheromone card, you likely have an infestation nearby and will need to make an insecticide application. Another way to figure out if you're going to have mealybug infestation in the following season is scouting fruit at harvest. If mealybug was present in your block in this season, you will find evidence of egg sacs and mealywags where the fruit is clustered, where the fruit is touching each other, or on the navel and calyx end. That is one way to tell if you're going to have mealybug infestation in the following year. This year, because we don't really know the seasonal phenology of citrus mealybug in the valley, we started monitoring a block in Ivanhoe using sticky traps to see if sticky traps can be used as a method to monitor citrus mealybug activity in blocks. And what we saw is sticky traps are pretty, effective in catching things if they're there. We started seeing crawlers in August, and then in April, first week of April, and in second and third week of April, most of the trap catches were females. And again, late in April, we started seeing more crawlers, and we're still seeing crawlers through June 1. Where we're trapping citrus mealybug males using pheromone traps, we saw male flights peak in that week of 1st of April. And this kind of ties back to what we're seeing in the sticky traps, that, once the females are mated, once the second star females are mated, we're starting to see adult females on the sticky traps after the males emerge, and the crawlers following that. So this, it seems like, is the first generation of citrus mealybug this season, starting as early as April. We are continuing to monitor these blocks using sticky traps and male trap cards through the season. We also did some trials to test what insecticides are effective. We tested several insecticides. We tested Center, Exidol, Azadirachtin, Dimlin, Beleaf, Lime Sulfur, SL, Senstar, Cefina, 2 different concentrations of Cefina, Cevanto, Movento, Steam, and compared it with the untreated control. The treatments were applied on 29th of March, and we monitored populations 7 days after the treatment, 14 days, 21 days, and 28 days after the treatment. And what you can see in these graphs is pretty much all the treatments reduced population in first week compared to control. But starting second week, in some of the treatments like Exidol, Dimlin, Beleaf, the population starts building up. Whereas in SL, we didn't see population, we saw reduced control up to 4 weeks. That was the only treatment where we saw population reduce up to 4 weeks. Whereas in other treatments, the population start coming back in 4 weeks. We're going to be repeating some of these trials with June populations and doing multiple application on the same tree. Also look at different rates of the insecticides that are effective in this season and in the coming season, so we have a better understanding of what works on citrus mealybug. So to conclude today's webinar, the take-home message from today's talk is that citrus leprosis is an emerging threat. This is a vector transmitted disease and can be prevented by regulating the transport of plant materials that may harbor vector and also vector management. When it comes to best management practices, June to August, September is the time to monitor and manage California Red Scale, Citricola Scale, and Fluorosphere Beetle. And also that citrus mealybug is a rising concern for citrus growers, and we are conducting studies to understand seasonal phenology and management for this important pest. And with that, I would like to thank you all, Beth especially, for mentoring me in this role and generating all the information most of the information that I shared with you today. My team, my group, Booth Ranches and Georgina Reyes for their collaboration in the Mealybug project, and Citrus Research Board for continuing to fund my research, and the UC A&R for everything. And thank you for participating in the poll questions and also listening to the webinar. If you have any questions.
So we do have a few Q&A questions, but
Okay.
we're going to put those up, but I know we're running a little bit over time. So I'm going to go ahead and put the survey link in the chat for anyone that needs to jump off. You're welcome to stay for the Q&A questions. So the first question we have, does Leprosis prefer colder or warmer temperatures?
That's a good question. I think the answer, how I would like to answer that question is, I'm not sure what weather conditions Leprosis proliferates in, but the vector mite definitely prefers warmer temperature and warmer and drier temperature. So I would think Leprosis also proliferates more, by extension in an area where vector is present, so warmer temperature.
So in blocks where mating disruption is implemented, how should pheromone cards be placed? Every 10 acres, near known populations, etc.
So pheromone traps are used to monitor the population. You would use a similar method, 2 traps every 10 acre towards the center of the block. But if you know hotspot, usually you'd put that around the hotspot where, if you have a hotspot in your block, either it's a border tree, border rows, or one corner of the block, then that's where you want to put at least one of your traps, and 2 more traps for every additional 10 acres.
So is the control of ants important for control of these scale and mealybug pests? Do ants farm these species?
Yes, it has been known and shown in several researchers that Argentine ants farm several mealybug species once, because they feed on honeydew produced by mealybugs. They will literally transport mealybug from one area of the tree to another. It has been shown in vineyards for ants to carry the mealybugs and carry them from one vine to another vine. So yes, to answer your question, ant control needs to go hand in hand with mealybug control.
We have a few in the chat too. So Enrico asked, you had Safina on the mealybug control graph listed twice. Why are the results so different?
We tested 2 different concentrations of Safina in that trial, 14 ounces and 28 ounces, which is why the results look different.
And then has any new research been done on Cryptolemus to control mealybug? Not having lures been to control ants prior to releasing makes control more difficult.
Can you read the question once again, Carolyn?
Has any new research been done on the Cryptolemus to control Mealybug? Not having lures been to control ants prior to releasing makes control more difficult.
Yes, we are working. We actually have a treatment going on where we release Cryptolemus in a block where, with known Mealybug population, we haven't finished collecting the data. So that's there is a research going on to look at the effects of Cryptolemus. And the second part of the question, please, again.
There was just one question on that one.
Okay. So yeah, the research with Cryptolemus is going on. And this year, most of the research we're starting is sort of trying to figure out what we need to do and how we need to focus. So we, all insecticide products, we looked at the label rate, highest label rate that is recommended. Next year, depending on what insecticide does better, we're going to look at different rates, maybe work with different volumes of water and on different citrus varieties.
So thank you everyone for attending the first webinar of our 2022 webinar series. Make sure that you mark your calendars for the rest of the webinars in our series. Next week, we'll be joined by Dr. Georgios Vilakis to discuss the Citrus Clonal Protection Program. And thank you to Sandipa and Greg Duhan for sharing their valuable information with us today. Thank you, everyone.